Welcome to part 20 of this video on the immune system. Okay, so we're currently discussing B cell production, and the point that we've got to so far is that the lymphoid progenitor cells have been produced and they're staying in the bone marrow. And we now want to understand how these lymphoid progenitor cells are going to design their own immunoglobulin heavy chain and their own immunoglobulin light chain. Specifically, they're going to design the variable immunoglobulin domain of the immunoglobulin heavy chain and the immunoglobulin light chain so that they will produce immunoglobulin molecules of their own design with their own specific antigen binding region. So we're now going to discuss this process, and we're going to discuss it for the immunoglobulin heavy chain and then the immunoglobulin light chain. So we'll begin with the immunoglobulin heavy chain, and I hope that by discussing the genetics of the immunoglobulin heavy chain, it will help you further understand the fact that we have these different classes of immunoglobulin molecules that a single cell is capable of producing. Okay, so if I just get a new sheet of paper here. So this is a absolutely classical example of VDJ recombination, far more classical than the examples that we've seen so far of the T cell receptor alpha chain and the T cell receptor beta chain. If you remember, in the T cell receptor alpha chain and the T cell receptor beta chain, we didn't really have diversity gene segments performing the same role as the variable and joining gene segments. I mean, in the alpha chain, we didn't have diversity gene segments at all. In the beta chain, we did have diversity gene segments, but they weren't performing the same role as the variable and joining gene segments. It wasn't quite the same. We didn't just pick a diversity gene segment in the way that we picked a variable and joining gene segment. In the case of the immunoglobulin heavy chain, you are going to have diversity gene segments in the exact same role as variable and joining gene segments. Okay, so we're now going to have a look then at the immunoglobulin heavy chain locus. So the first question is, what chromosome is the immunoglobulin heavy chain locus actually on? And the answer is it's on the same chromosome as the alpha chain of the T cell receptor, specifically chromosome 14. So of course you'll have two chromosome 14s, and therefore you'll have two immunoglobulin heavy chain loci, and the story is exactly the same as for the alpha and beta chains of the T cell receptor. We will use one of the immunoglobulin heavy chain loci, and if our VDJ recombination goes correctly, we'll just ignore the other one. We won't bother doing anything to it. If we don't bother performing VDJ recombination on it, then it won't ever actually produce any immunoglobulin heavy chains, and therefore it won't cause a problem. So it's not a problem just leaving it alone. If, however, the VDJ recombination goes wrong with our first immunoglobulin heavy chain locus, then we will move on to the immunoglobulin heavy chain locus on the homologous chromosome. Okay, so the next thing I want to do is actually show you what this immunoglobulin heavy chain locus looks like in the germline form, which will of course be the form that it's in the lymphoid progenitor cells before we've actually begun trying to design our immunoglobulin heavy chain. Okay, so let me show you this. So, as always, these two parallel lines will represent the double-stranded DNA, and what I'm going to do is I'm going to show you the different gene segments on my immunoglobulin heavy chain locus by just drawing little boxes around certain sections. Okay, so let's actually begin this. So, at the beginning of the immunoglobulin heavy chain locus in the germline form, we're going to have, as always, a great big cluster of variable gene segments. So let me put them here. And I'm not going to give you the numbers, the exact numbers of how many variable gene segments and diversity gene segments and joining gene segments that we have in the germline form of the immunoglobulin heavy chain locus. One, because as I've stated previously, it's not as important as the fundamental concept. Secondly, because the numbers are liable to change. So focus on the concept rather than the specific numbers, but know that the numbers will be between 1 and 100, so that's the sort of numbers, the sort of scale that we're dealing with here. So we're not dealing with numbers that are in like thousands or millions. Okay, so you'll have a cluster of variable gene segments here. all of which you could potentially use to actually produce your immunoglobulin heavy chain. So let me just colour these in here. So I'll colour them in red. So what the first variable gene segment here, the second variable gene segment here, all the way up to the last variable gene segment there. So this is a cluster then of variable gene segments. Okay, so as always in VDJ recombination we'll just be choosing one of these to actually use in the coding of our immunoglobulin heavy chain. Okay, after the variable gene segments, what we will have is a cluster of diversity gene segments, and here, as I said previously, the diversity gene segments are going to have an identical role to the variable and joining gene segments, i.e. we're going to have a cluster of them, separated of course by introns, just like we have a cluster of the variable gene segments and the joining gene segments. So this is a whole cluster of diversity gene segments, and I'll colour these diversity gene segments in blue here. So here's the first diversity gene segment, here's the second diversity gene segment, and here's the final diversity gene segment. And again, in the process of VDJ recombination, we'll just be selecting one of these diversity gene segments to actually use in the coding of our immunoglobulin heavy chain. Okay, so there's a cluster of diversity gene segments that you could potentially use, and then right at the end we'll now have a cluster of joining gene segments. So here is the first one, here is the second one, and then we'll go on all the way up to the final one here. So this is our cluster of joining gene segments. Okay, so I'll colour these in green here. Right, so you might think, well, that's going to be it now. But of course it gets more complicated than this, and in fact actually this is the simple bit. The next bit is the far more complicated bit, because we need to put in the constant gene segments, and unlike the alpha chains and beta chains of the T-cell receptors where the constant gene segments were very, very simple, in the case of the immunoglobulin heavy chain though, because we know that the constant gene segments are actually going to be a nightmare, they're going to be incredibly complex, and indeed they are. So bringing back up the previous piece of paper here, where we were discussing all of the different classes of immunoglobulin molecules, remember we said that these different classes of immunoglobulin molecules, they differed by the fact that if you were to look at the heavy chains in these immunoglobulin molecules, they would have different constant regions, and we gave the names for all the different constant regions of all these different immunoglobulin molecules. Okay, so in the immunoglobulin heavy chain locus, you're going to have all 10 of these different constant gene segments afterwards, and you're going to be able to use any of them basically to produce the immunoglobulin heavy chain. Okay, so unfortunately the order is not simple, so I'm now going to show you the order. I give you the order not because it's particularly important that you know the order, but just to absolutely make this concept crystal clear. So here we go, I'll draw 10 little rectangles out firstly, so 1, 2, 3, 4, so these are just going to represent the constant gene segments, all of course with introns in between each other. So this is 6, 7, 8, 9, and then finally the 10th one here. Okay, right, so let me now show you the order. So you should at least know the first two. So if you're going to memorize a little bit of this order, just memorize the first two, because these are actually important that you realize that these are the first two. So the first one is the constant mu gene segment. So this is the gene segment which, if you use it to code for immunoglobulin heavy chain constant region, will make you a heavy chain that will participate in an IgM molecule. So if you make an Ig molecule from those heavy chains where the constant region was coded by the constant mu gene segment, then it will be called an IgM molecule. Next one up is constant delta. Okay, so if you were to use that constant gene segment rather than the constant mu gene segment, then the heavy chains that you'd actually produce when you put them into an immunoglobulin molecule, that immunoglobulin molecule would be called an IgD molecule. Okay, continuing on now, and as I say, it's not particularly important that you know this order. What's important is that you completely understand the concept. So I give you the order just to make it concrete, rather than because I think it's important that you go away and memorize this. Okay, so next up is constant gamma 3. Then it's constant gamma 1, and the only reason I'm alternating between up and down is just for space reasons. So constant gamma 3, then constant gamma 1, then constant epsilon 2, then after that constant alpha 1, then after that constant gamma 2, then after that constant gamma 4, after that one it's constant epsilon 1, and then the final one that's left over, which is alpha 2, constant alpha 2. And I will just try and color code this just to make it look a little bit more interesting. So we'll have constant gamma 3 here in orange. So if you were to use that one to make your immunoglobulin heavy chain constant region, then those immunoglobulin heavy chains, when they go into immunoglobulin molecules, will now produce IgG3 immunoglobulin molecules. Here is constant gamma 1 there in red. Constant epsilon 2 follows constant gamma 1, and I'll colour this in pink here. Constant alpha 1, I'll colour in yellow. We're going to have to repeat the colours because I've run out. Constant gamma 2 we'll have coloured in blue here. Constant gamma 4 we'll have coloured in orange, like so. Constant epsilon 1 we'll have in red here, like so. And oops, forgot to underline it. And I forgot to underline also constant gamma 4, so I'll just come back and do that. And then finally constant alpha 2 right at the ends there in green. So I hope you now have a better understanding of that. These variable diversity and joining gene segments will choose one of these, one of these, one of these in the VDJ recombination. And those 3 gene segments together, they will then code for the variable immunoglobulin domain of the immunoglobulin heavy chain. After that, we can then choose one of these constant gene segments to use in the coding of the constant region of our immunoglobulin heavy chain. And depending on which one we produce, well, which one we use, we will end up with a different immunoglobulin heavy chain. The structure will change. You will have a different primary sequence of amino acids, and you will have a different immunoglobulin heavy chain. So no matter which one of the constant gene segments you use, you will always have the same variable region, however, and therefore it won't change the antigen binding site. The other key thing for you to understand is that in VDJ recombination, you're not going to do anything with regards to these constant gene segments. So VDJ recombination is just going to design the variable region of the immunoglobulin heavy chain. It's not going to mess around with these. Okay, the next thing that I want to tell you about is that each of these constant gene segments, it will have a section in its DNA, in its sequence, it will have a section right at the end which will code for the portion of the immunoglobulin heavy chain which can implant into the cell membrane. I know I've said this again before, but I just want to really hammer the point home. So each one of them will have a section right at the end of the gene segment which will code for the portion of the immunoglobulin heavy chain which will implant into the cell membrane. And this will be transcribed, and then it can either be spliced out or left in, and that will then result in the secreted or membrane-bound form of each of the different types of immunoglobulin molecule. The other thing that I want to stress, because it's really important and I want to make sure it's absolutely clear, is that when you actually produce an immunoglobulin molecule, the 2 heavy chains that you use will be identical. So you cannot use a heavy chain where you have used the constant delta gene segment to code for the constant region of the heavy chain with an immunoglobulin heavy chain where you've used the constant mu gene segment to code for the constant region. They have to be identical. Okay, that's a no-goer. You can't have chimeras like that. Okay, right, so here is the germline form then of the immunoglobulin heavy chain locus. What I now want to go through is VDJ recombination of this. So what's going to now happen in our lymphoid progenitor cell in order for it to design its own immunoglobulin heavy chain? So you should be able to tell me, because it's exactly the same as for the alpha chains and the beta chains of the T-cell receptor. So what you're going to do is you're going to select a variable gene segment, you're going to select a diversity gene segment, and you're going to select a joining gene segment. So each one of the lymphoid progenitor cells will pick a variable gene segment, a diversity gene segment, and a joining gene segment that it's actually going to use to make its immunoglobulin heavy chain gene. Then what will happen is it will cut out the DNA between these chosen gene segments. So again, because it's the easiest way to do it on this picture, let's say that I pick the second variable gene segment here, so V2 here, and the second diversity gene segment here, so D2, and also the second joining gene segment here. It's just the easiest way to do it. So we'll pick this one, and this one. Let me now illustrate what will happen in VDJ recombination in these lymphoid progenitor cells. So what will happen is you'll cut out all of the DNA between your chosen variable gene segment and your chosen diversity gene segment, and you'll also cut out all the DNA between your chosen diversity gene segment and your chosen joining gene segment. Okay, so you'll cut out all of that. You do not make any changes at this stage to the constant gene segment region over here where all of the constant gene segments are. Okay, that will come much later. So let me now draw out what the immunoglobulin heavy chain locus will look like in one of these developing B cells that has undergone the process of VDJ recombination as I have just described. So now after VDJ recombination, what's this going to look like? Okay, so we're now going to have our variable gene segment next to our chosen diversity gene segment. So if I draw this out here, like so, we will have our first variable gene segment, because that hasn't been cut out. So in this case, because we chose the second variable gene segment, the only one upstream of it was the first variable gene segment. If we'd chosen one much further along, then of course all of the variable gene segments that were upstream of that would still be there. They'd still be upstream of it. But in this case we've just got one shown there. So there's the variable gene segment that was upstream of the chosen variable gene segment. Okay, in red there. Here is our chosen variable gene segment, and now it's going to be next to our chosen diversity gene segment, and our chosen diversity gene segment is then going to be next to our chosen joining gene segment. So colouring this in, our chosen diversity gene segment is here in the middle in blue, and our chosen joining gene segment is on the end in green, like so. Then of course, just like was the case with the T-cell receptor alpha chain and the T-cell receptor beta chain, all of the joining gene segments after the one that we had chosen are still actually there. They're still left in. So even though it would seem intuitive to now remove all of the joining gene segments that we're not using, that's not what actually happens. So all of the joining gene segments downstream of the chosen joining gene segment are still going to be here, so like so. So this is J3 if you like, and it will go all the way up to the final joining gene segment here. So I'll colour these in green like so. And then what you'll have is all of the constant gene segments still there. Now I'm just going to draw the first two because they're going to be really important for our discussion now. So I'm going to draw the constant mu gene segment and the constant delta gene segment. So here is constant mu and here is constant delta. So we'll have constant mu again in yellow and we'll have constant delta in blue. But all the other constant gene segments downstream of constant delta will still be there. I just don't want to bother drawing them for this picture. Okay, so this is what happens you will choose a variable gene segment, you'll choose a diversity gene segment, you'll choose a joining gene segment, and you'll recombine the DNA in this way. You'll cut out the intervening DNA. And of course, this portion here is now going to code for the variable immunoglobulin domain of our immunoglobulin heavy chain. What we now need to discuss is how does this gain all of its diversity, which is simple. It's the same answer as was the case with the alpha chain and beta chain for the T-cell receptor. And then I also need to discuss what's actually going to be transcribed when the RNA polymerase enzyme works its way along here. Okay, so the way then that we produce diversity is in two ways. So firstly, there is the concept of combinatorial diversity, which is exactly the same as it was in the case of the alpha chain and beta chain of the T-cell receptor. So we have got a huge number of different combinations of variable diversity and joining gene segments that we could pick here to create this little portion that's actually going to code for the variable region of our immunoglobulin heavy chain, and therefore that's one of the ways that all the different lymphoid progenitor cells are going to get a diverse arrangement of immunoglobulin heavy chains. So that's combinatorial diversity. The second answer, of course, is then we also have junctional diversity. So again, the places where you cut the DNA that you're going to snip out, it's not set in stone. There is not an absolute place where this cut had to be made. Okay, there's a rough region where it's going to be made, but it's not set in stone. So there are, when you actually perform this recombination event, there are different ways that it can actually occur. And that junctional diversity is going to mean that there's different amounts of DNA in between the chosen variable, the chosen and the chosen diversity, and the chosen diversity and the chosen joining gene segment. And that's also going to help to allow all the different lymphoid progenitor cells that we're producing to create their own unique region of DNA here that's actually going to code for that variable region of the immunoglobulin heavy chain. Okay, so that's combinatorial diversity and junctional diversity. Of course, the same problem with junctional diversity that we had in the case of the alpha chains of T-cell receptors and beta chains of T-cell receptors occurs here. And that problem is that you might end up reading the diversity and joining gene segments in an out-of-shift frame, in the wrong reading frame. And if that occurs, then VDJ recombination has failed. This will not produce a viable immunoglobulin heavy chain, and therefore you will have to go to the other chromosome 14 and you'll have to do VDJ recombination on the other chromosome 14 and hope that it works better there. And of course, if it fails on both of the immunoglobulin heavy chain loci, then the developing B cell will have to commit apoptosis. Okay, now what I want to talk about is what is actually going to be transcribed. So what's going to be transcribed is you're going to start at the start of the chosen variable gene segment and you're going to work your way down. Okay? The new question, because at the moment this just looks exactly like the problem that we had in the alpha chain and the beta chain of the T-cell receptor, but there's a new question here, and the new question is, do you actually transcribe all of these different constant gene segments? And the answer is no. And this is the reason that I've only shown the constant mu gene segment and the constant delta gene segment in this picture. So you transcribe from your chosen variable gene segment all the way up to the end of the constant delta gene segment. So this is what you're overall going to get. Here is the piece of mRNA corresponding to your chosen variable gene segment. Here is the piece of mRNA corresponding to your chosen diversity gene segment. And here is the piece of mRNA corresponding to your chosen joining gene segment. Then you'll have RNA that corresponds to all of the other joining gene segments with introns in between them, like so. Then you'll have your constant gene segments, and you'll have the mRNA for the constant mu gene segment there, and the constant delta gene segment there. But you don't go any further. Okay, so this is what would happen if the developing B cell was to now express its VDJ recombined immunoglobulin heavy chain locus. So let's say we had a lymphoid progenitor cell which was going to become a B cell. It performed VDJ recombination on one of the immunoglobulin heavy chain locuses that's on one of the chromosome 14s, and it was successful. And it's done it just like so, and we're now asking what will actually be transcribed, and this is the answer as to what will be transcribed. You'll get this as your pre-mRNA. Okay, so how will this developing B cell now actually produce the immunoglobulin heavy chain that it's designed? Well, what will happen is all of this mRNA here will most definitely be spliced out. So all of this will be spliced out. So splicing will remove all of this. Okay, this portion of course we want to leave in because this is the portion that's going to actually code for the variable region of our immunoglobulin heavy chain. The question now becomes about the immunoglobulin heavy chain's constant region. Which of these two bits do we use? Do we just use both of them? Well, the answer is no. What you're going to do is splice out one of them. So if you wanted to use the constant mu gene segment, then of course you would splice out this bit. So you'd get rid of that intron there, and you'd get rid of the constant delta portion here. If you wanted to use the constant delta gene segments to code for your constant region of the immunoglobulin heavy chain, you'd splice out all of this bit. You'd get rid of the constant mu gene segment and that intron there, and then you'd attach the constant delta portion to this portion, and then you'd have your mature mRNA. So let me just show you an example of a mature piece of mRNA here. So an example, let's say we use the constant mu gene segment overall. So here is our chosen variable gene segment. Here's our chosen diversity gene segment, and here is our chosen joining gene segment, and then after that we'll have the constant mu gene segment here in yellow, but it could have equally well been the constant delta gene segment. So this would then be our mature mRNA, and of course we would then produce an immunoglobulin heavy chain that would produce IgM immunoglobulin molecules, because it would have used as the to code for the constant region of the immunoglobulin heavy chain, the constant mu gene segment. Okay, so initially then, these developing B cells, they are only capable of producing IgM and IgD molecules. This is really important that you understand this. The developing B cells are only capable of producing IgM and Ig delta because of the fact that transcription does not go any further. If transcription went all the way along to the end, it went all the way up to constant alpha 2, then of course you'd have your full range, and this developing B cell would now be able to express immunoglobulin heavy chains of all the different types, i.e. with all the different possible heavy chain constant regions, but it doesn't. Transcription only goes up to constant delta, and therefore actually these developing B cells are only capable of expressing IgM and IgD. So you might be wondering now, well, what's the point of all the other ones then if they're not actually going to be used? They will be used later on. You will see where the others can actually be turned on later on. So you can produce the other types of immunoglobulin, but it requires further modifications that won't occur until after B cell activation has occurred. So we'll see these ones again, but later on. Okay, so initially a developing B cell can only express heavy chains that are going to contribute to IgM or IgD immunoglobulin molecules, i.e. that have a constant region that is either constant mu or constant delta. Now, the other thing to say is that each of these constant mu and constant delta gene segments, as I previously said, it will have a portion right at its end. So I'll show this for the constant mu and the constant delta here. So these portions right at the end of the gene segments, those will be the bits coding for the integral membrane portion, the bit that can actually span the cell membrane. And at the splicing stage, where you're going from mature mRNA to from pre-mRNA to mature mRNA, you will have to decide, do you want to include that bit? So you could splice it out, and then you'd end up with an immunoglobulin heavy chain that would contribute to a secreted form of immunoglobulin, or you could leave it in, and then you'd end up with the membrane-bound form of the immunoglobulin. Initially, these developing B cells, they are just going to produce the membrane-bound bit, so they're going to leave this in, as I've shown here, and they're therefore going to end up with immunoglobulin heavy chains that will contribute to membrane-bound immunoglobulin molecules. Okay, so where have we got to overall from here? We have seen the VDJ recombination event that will occur on the immunoglobulin heavy chain on chromosome 14 in our developing B cells. Okay, we have also understood that after this, the developing B cells will be able to express their immunoglobulin heavy chain, but they only express it in the IgM or IgD form, and they only express it in the membrane-bound form of those two forms. So they will produce membrane-bound immunoglobulin M and immunoglobulin G heavy chains. So far we haven't seen yet whether they, we haven't seen yet the designing of the light chain, and therefore they won't have produced that yet, so they won't be producing full immunoglobulin molecules yet, but so far they will now be able to produce these immunoglobulin heavy chains with their specific variable region that is unique to that developing B cell, and of these two different types, the IgM type and the IgD type. Okay, so we'll have a break here, and in the next video what we will do is look at VDJ recombination for the light chain, so the designing of the light chain of the immunoglobulin molecules.